Hello, everyone. My name is David Bogumil. I am a postdoctoral research fellow at the University of Southern California's Keck School of Medicine, and I'm going to be covering lectures 3 and 4 with you guys. So today we're going to start off with lecture 3, Introduction to Polygenic Risk Scores. And as we go through the slides here, I will have some citations in the slide notes and also on the screen in case you guys want to look into any of these specific concepts or topics in a more detailed way. So there's two major sections to this lecture here. We're going to go over the basic concepts of polygenic risk scores, so kind of what type of information is being captured in a polygenic risk score and how we choose to select different pieces of genetic information to go into the polygenic risk score. And then second, we are going to kind of go through a more hands-on calculation of how to do a unweighted and weighted polygenic risk score. And I have on the bottom of the screen here a reference to an article which goes into a bit more detail about polygenic risk scores, some of the methods and caveats. So I highly recommend checking this out if you guys want more detailed information on some of what we're going over today. All right, so in order to understand what we are measuring in a polygenic risk score, we need to first revisit the idea of a single nucleotide polymorphism. Now, commonly polygenic risk scores are built up of a bunch of these single nucleotide polymorphisms summed into a single score. In the examples we're going over here, we're just going to focus on these SNPs. However, it is important to keep in mind that some polygenic risk scores may also use other type of genetic variation in addition to SNPs, such as insertions and deletions. But just to focus on polygenic risk scores for what we're going over today, and to recap, so remember, polygenic risk score, it's just a more modern unit or measurement of genetic variation. And really what we're looking at with a single nucleotide polymorphism is just a single base pair on a piece of DNA. And that's what I have illustrated here on the right hand side. So we have two copies of DNA, all of us do, and we have an allele or a base pair at each location. So here in this example with the highlighted or the circled base pairs, we see we have C at one location and T at the other. And when we're measuring and looking at the single nucleotide polymorphisms, we're just concerned with one side of the DNA strand because we know what the complement will be. So here, the two alleles that are possible for this given location are C or T. And most commonly, when we're looking at a given location on the DNA, there are two alleles. So that's exactly what we see here. It's possible for this person to have a C or T. They could have two C's, two T's, or a C and a T, or a T and a C, which would be the same thing. Now, as we move into kind of understanding how a polygenic risk score is built, it's important to think back again to, what information are we trying to capture with this polygenic risk score, right? So we have the base pairs measured on the DNA, right? But what does that genetic variation on the DNA really mean, and how does that correspond to somebody either getting or not getting a specific disease outcome, right? And when we're looking at a specific variant on the DNA, right, we're measuring in association with an outcome, we're looking to measure some type of genetic variation that results in different proteins, right, through this process of transcription, translation, and finally protein folding, right? So as we get towards kind of these later stages of going from DNA to protein, right, we're going to be altering the biology, which can either increase or decrease the risk of disease. And what we're doing in the polygenic risk score, right, is we're going to be looking at a number of variants on the DNA and kind of trying to sum up all of these into a single score. So, in order for us to kind of move through this lecture from, understanding generally what a polygenic risk score is and how it's built into actually calculating one and how looking at how we assess performance, I thought it might be nice to kind of give us a structure to follow through here. And we're going to start up here in the upper left at the analysis phase for GWAS discovery. And this is where we're conducting the GWAS analysis, or other people are conducting the GWAS analysis, and they're looking at all the genetic variants, right, across our chromosomes and determining which ones are associated with disease, right? And once the GWAS is connected, then we get our summary statistics. And these GWAS studies, commonly logistic regression and linear regression is used, whether we're looking at a discrete or continuous outcome. But it is important to keep in mind that there can be a variety of outcome types besides even just discrete, like logistic regression having two levels. And there can also be a large variety of study designs that give rise to this data too. Commonly for GWAS studies, we see case control, but it's possible to use data from biobanks and things like that, which don't necessarily use these study designs. All right, so like I said, for the first section, let's first look at GWAS discovery and summary statistics, which will lead us into identifying the variance for our polygenic risk score and the calculation. So down here I have the results, which would come from a genome-wide association study. Right here on the right-hand side, you guys can see the Manhattan plot, which you're all probably fairly familiar with, where we're basically going through each base pair on the DNA, and we're looking to see do the alleles, or the variation in the alleles at that given location, is it associated with an outcome of interest, whether that be a cancer like prostate or breast cancer, or cardiovascular outcomes, anything. And we generate the results from the GWAS studies after we go through and we do the association analysis for every single base pair, and then we plot the results in this Manhattan plot here, and we have on the y-axis a transformed version of the p-value in order to look at the significance of each of these statistical tests, where we're looking at the association between each of these SNPs and the outcome of interest. And the variance that pass this threshold that is predetermined, that we set beforehand, this genome-wide significance threshold here, which is indicated by this dotted line, the variants that pass that are what we consider to be significantly associated with the outcome of interest. And those represent ones where variation for those SNPs is going to determine the risk of the outcome and would likely provide valuable information for the inclusion in a genetic risk score, right? If we're wanting to kind of sum up all of these significant hits into a single score. Now, one important thing to keep in mind, and I highlight this on chromosome 19 over here, is that due to the linkage disequilibrium and association between adjacent genetic material, we'll often be in situations where we don't know if we're identifying the actual true causal SNP, where the alleles change in that SNP, it results in differing disease risk, or if we're actually just measuring an adjacent SNP, which is kind of acting as a proxy or tagging the true causal SNP. And the example you can see here, I kind of zoom in a little bit on chromosome 19, is, it is possible that we have this genotype SNP here on the right, but then so many base pairs prior is the actual disease risk SNP, which we may or may not actually have as results in our genome-wide association study. But for the most part, it's all right if we don't have that true causal SNP, as long as we have a good proxy SNP, which measures disease risk for that region. Now, just to recap, as I was mentioning before, these results here in this Manhattan plot come from the genome-wide association study. And each one of these hits, in this case, I circled the most significant hits, right, have a effect size, a beta associated with them, and a p-value also. And over here on the right-hand side, I show you the with this notation, we're saying what is the log odds ratio for the genetic variation at this position. So this is a chromosome and a position. And then we have our alternate allele and our reference allele there. And then we have our beta value here, which is 0.05. And for this GWAS, which you guys are probably familiar with, we're just implementing this model down here, testing each one of those variants. And then these significant variants from the GWAS studies are commonly what we decide to include in our polygenic risk score, because they significantly determine disease risk. So we have the results from our genome-wide association study, and these are frequently, large pooled meta-analyses and things like that. And we're frequently just looking at the summary statistics from these studies, and then gathering that information, seeing what variants are significantly associated with outcome, and using those to build a polygenic risk score in our separate data set. And so here we're getting the summary stats, and then we're using that in the polygenic risk score calculation. And once we go through that procedure in our sample, we will get a new variable for everybody in our data set, which will be their polygenic risk score. So here we can look back at our Manhattan plot from the genome-wide association study, and we have our results on the right-hand side of the screen for what the effect sizes are for all the variants. And then down here, I am showing you the polygenic risk score data set. So let's say this is like a data set of 500 people we have collected in a study, and we want to calculate their polygenic risk scores. Now we have our results from the genome-wide association study, and what we need to do is for our data, for our independent separate data set, is we need to identify the genetic variants in our data by frequently we will just subset all of the genotype data we have for our sample. And this data is frequently held in a format which we call dosage, where over here, as I mentioned earlier, we have our chromosome number, then we have the position within the chromosome, then we have an alternate allele, and then we have a reference allele. So here the alternate is G and the reference is T. So that 2 means that this individual has, this first individual, let's say they're individual 1, has 2 copies of the G allele, right? And here if we were to calculate a weighted polygenic risk score, we're just getting the number of risk alleles they have for that genome-wide significant loci, right? That location, and we're seeing how many risk alleles do they have, and then we're multiplying that by the effect size from the GWAS study, the beta from the GWAS study, and that will give us a kind of weighted risk for that allele. And the reason why this is done is because there are certain associations which will have a much larger effect size. For example, in these results here, we see the second to last variant here has a beta value of 0.11, right? So there's a stronger association between having the risk allele and risk of disease, right? So when we look at this variant here that ends in 824, right? So that's down here, it ends in 824. This person has basically a dosage value of 2, meaning 2 copies of T, of the risk allele T, right? When we put that into the polygenic risk score, right, because we're going through and we're doing this kind of um, uh, multiplication between an individual's dosage and their weight for each of the variants. When we're doing that for this variant here, right, they have their dosage of 1.99 and we're multiplying it by 11. This gets, in the end, this genetic loci here, the SNP, gets a higher weight in importance in the final score because the effect size from the GWAS study, right, which is 0.11, is just larger, right? So it has more of an impact in determining disease risk. But basically, this process we go through, right, for each of the variants in our data set that are from the hits in the genome-wide association study. We find them in our data set, and then we just multiply an individual's dosage value by the weights from the genome-wide association study, and then sum those up, and that gives us a polygenic risk score. So here I have the example for the first individual in our data set, individual 1, and we're going through. They have a dosage value of 2 over here, and then we're multiplying that for the SNP, right? For the effect size from the corresponding um, loci from the GWAS study, that's 0.05, right? And we just go through and we do this for each one of the variants, right? Variant 1, 2, 3, 4, 5. This is 1, 2, 3, 4, 5, right? And then we sum them up into a final score, which represents the polygenic risk, right, for this individual, person 1. And then you could imagine this would basically just be a new variable here at the end, PRS, and that would be 0.4. And we just go across and do this for all the individuals. And, if we were doing this in a statistical program, we would just have code to do this. So with that new polygenic risk score variable that we've calculated, we can plot it as a histogram or just examine it as a distribution. And we expect somewhat of a normal distribution, although in some cases prior to using the polygenic risk score in analysis, we may put it through standardization. Here I show a little bit of the results from an analysis I'm working on in the multi-ethnic cohort, where we have the polygenic risk score calculated for 3 different phenotypes, breast cancer, colon cancer, and prostate cancer. And then we do this calculation across different racial and ethnic groups based on how people self-reported when they entered into the cohort. And as you can see, similar to the previous slide, right, we have fairly normal distributions here, and we do see some variation in the polygenic risk score statistics across populations. After we have our polygenic risk score calculated for our subsample, we then want to evaluate it to see whether or not this score, which kind of sums up the risk variance identified from prior GWAS studies, whether it actually captures the disease risk in our sample, right? So we need to use the score in regression models in order to assess its performance and capture that information in statistics. As I mentioned before, the type of model we use depends on the outcome, whether or not it's continuous outcome or dichotomous outcome. And that would determine whether we just use linear regression or logistic regression. And then in these models, we frequently will adjust for the same variables which were used in the genome-wide association study itself, even if we didn't conduct that genome-wide association study with our data. So here we're going to adjust for variables like sex, age at sample collection, and principal components as a means to adjust for population stratification and relatedness within the sample. Depending on the model type we use, whether it's linear or logistic, that will determine some of the statistics that we will get access to for regardless of if the model has a continuous outcome or a dichotomous outcome, we can always assess the beta value for the polygenic risk score and the p-value for the polygenic risk score, just to see if it is significantly associated with the outcome of interest. And then for both linear and logistic regression, we can look at R-square correlation statistics in order to see how much variation in the outcome can be explained by our polygenic risk score. And then lastly, and fairly commonly for logistic regression models, we'll also look at AUC statistics, area under the operator-receiver characteristic curve, in order to identify how well the polygenic risk score could classify individuals into cases and non-cases. Now, for the R-square and for the AUC also, frequently what is done is a base model will first be fit that does not include polygenic risk score as a variable. And then the R-square and the AUC statistics will then be compared against a full model that additionally includes polygenic risk score as a variable. And the reason why is you can imagine that in the base model, right, if we're looking at the R-square statistic or AUC, the base model, these variables, sex and age, right, are likely to be significantly associated with the outcome. Let's say we're looking at colon cancer as the outcome, right? If there's any sex differences in colon cancer, and if older people get colon cancer, then both sex and age will be significantly associated, right? And we may get an R-square or an AUC that is not zero, right, or is not null. So for example, here in our first model, we may get an AUC of, let's say, 0.59, right? And then in our full model, we could get an AUC of, let's say, 0.65, right? Here we would see that the additional inclusion of the polygenic risk score, it does contribute to determining who is a case or not relative to just including sex and age in the model. So I just mentioned this here. This isn't something that really needs to be concerned about for the beta and p-values. Normally those are just pulled out of the full model, adjusting for all the terms. But when you want to look at an R-square, or an AUC, frequently you will want to compare models. To recap this whole process, we start with the genome-wide association study, where we identify variants that are significantly associated with the outcome of interest. Now, we may conduct the genome-wide association study, or we may find genome-wide association study results off of databases or things like that. And then we use the results from that to estimate or calculate a polygenic risk score in our own sample. And this sample would likely be different from the sample in which the GWAS was conducted in. Frequently, it's a smaller independent sample. And we use those summary statistics along with the dosage information, which determines how many copies of risk alleles people have in our sample. And we get the dosage and we multiply it by the coefficient or the beta from the genome-wide association study, and then do that for each of the genetic variants and sum them all up into a single score, and that gives us the polygenic risk score, right? And this is a new variable we can look at in our data set. We can look at it and examine the distribution, just calculate descriptive statistics on it. And using that as a new variable in our data set, we will then want to evaluate, is it associated with the outcome of interest, right? I mean, the polygenic risk score is supposed to be a sum of common genetic variants that are associated with the outcome of interest, right, as identified in the GWAS study. So ideally, when we evaluate the polygenic risk score in our sample, it will be associated with the phenotype of interest. So that is commonly done through putting the polygenic risk score into linear regression models and fitting those regression models with our outcome, the same outcome which they used in the discovery in the genome-wide association study. And we then assess the model performance in a number of ways. We'll look at the beta coefficient for the polygenic risk score. We'll look at the p-value for the polygenic risk score. And then we'll look at the overall model performance, the R-square and AUC statistics. But frequently, we need to compare, that kind of reduced simple null model that doesn't include the polygenic risk score in it against a model that has polygenic risk included in it to see how much more can the polygenic risk score determine disease status in our sample beyond just the simple covariates we're including, like sex and age. The example that we've went over in this lecture represents the most common form of polygenic risk score calculation, which is a weighted polygenic risk score. Now, it is important to keep in mind if you were to conduct polygenic risk score analysis or just evaluate how well a polygenic risk score works in a sample you have, it's important to keep in mind a number of considerations. And I briefly list these here, but I do recommend going back into some of the articles which I have in the slide notes or at the bottom of the slides in order to become more familiar with these if you are going to carry out some of these analyses. But just to mention a few here, first we have polygenic risk score standardization. Now, frequently, polygenic risk scores are standardized in some way before they are entered into a regression model to be evaluated. Now, the standardization commonly is just like how you would standardize any other continuous variable prior to conducting a regression on it. The second consideration is the independence of single nucleotide polymorphisms included in your polygenic risk score. Now, it is important that each single nucleotide polymorphism that goes into your score contributes unique genetic information, right? If you have a polygenic risk score with a bunch of SNPs that are in close association with each other, then whatever information is coming from that genetic region is going to be too overweighted, right? So what you can commonly do if you're developing the polygenic risk score yourself is examine the association, pairwise association between all of your single nucleotide polymorphisms to make sure they're each contributing unique information to your score. The third consideration is coding of risk alleles. In the examples we went over here, all of the um, all of the alleles were coded in a way where we did not need to flip them, and this idea basically corresponds to having an alignment between your data and the GWAS data in your coding of what a risk allele is, right? So if in the GWAS data for a given SNP, they know T to be associated with increased risk of disease, in your data set, you want to make sure that T is coded to also correspond to increased risk, right? And you need to make sure that this is done for all of the variants in the genetic risk score. Fourth is sample overlap between discovery and polygenic risk score evaluation data sets. Ideally, the sample in which the genetic variants were discovered to be included in your polygenic risk score. Now, this is like the GWAS data set, basically. Ideally, that is independent and separate from the data set in which you're evaluating the polygenic risk score in. And then second to last is participant relatedness. Just like with any regression, you want independence between all of your participants or your observations in the data set. And if you don't have independence between your observations, you will get an inaccurate measure of statistical significance for your variables. And the very last is polygenic risk score weights. Like I said, in this example that we went over here, we used a weighted polygenic risk score where the selected set of SNPs were GWAS significant SNPs, and the weights came from the GWAS. However, there are a large variety of methods for polygenic risk scores, some even which use all genetic material or genetic variation that's available, like a genome-wide genetic risk score. However, the weighted polygenic risk score, which we went over today, is by far the most commonly implemented one.